I am Dr. Zia Ansari. I am teaching today DNA replication. What is DNA replication? A reaction in which daughter DNA are synthesized using the parental DNA as a template, that is called as DNA replication. For this replication, there is the transferring of the genetic information to the descendant generation, which a high fidelity. What is the DNA replication? See, these are the DNA. You know the DNA is a double helical structure. See, these are the double helical structure of the DNA. These are coiled together. There is presence of the nitrogen bases as well as the sugar as well as the phosphate backbone. They are united together. See, this one will be unwound, and because of the unwinding, these two strands get separated. If these two strands that will be get separated, each of these strands they will be having the capacity to forming into the new strand. So like this, on these two cases, when this one will be unwound, these two will be formed. See, this DNA replication, there is the types. Number one will be called as semi-conservative DNA replication. Semi-conservative. Second one will be called as conservative. And the third one will be called as dispersive. See, these are the three types of DNA replication. See, for this, there is the two different types of the DNA replication in prokaryotes as well as the eukaryotes. These two replication will be occurred. What is the mechanism of DNA replication? DNA replication is catalyzed by DNA polymerase, that is the enzyme which needs the RNA primers. RNA primers, so these are the RNA primers. These RNA primers synthesized on DNA strands. The DNA polymerase adds nucleotides to the 3' end of the growing strands. So the DNA strands, number first type, that will be called as semi-conservative. Half of the parental, this is the example of semi-conservative, half of the parental DNA molecules is conserved in each new double helix paired with a newly synthesized complementary strand. This is called as semi-conservative. See, this one will be parental, another one will be new. One will be parental, another one will be new. This is called as half conservative, semi-conservative. See, for this, the replication, there is the three stages. Number one will be called as initiation. Initiation, they occurs at the origin of the replication. They separate DNA primer synthesis. Second one will be called as elongation. Elongation, they involve the DNTPase based on complementary of the template strand. They are addition of the new nucleotides and they forms the phosphoester bonds, correct the mismatched bases extending DNA strands. And the third one will be called as termination. After completion of this, there will be stop the growth, that will be called as termination. This is the three stages of replication. See, for this replication, there is the for this process, there is the first one will be called as, for the replication, first one will be called as unwinding of DNA. False, the replication 4. If these strands will be unwind, there is formation of the 4. The second one will be called as primase. In this primase, they will be synthesized the RNA primers. There is the continuation of the synthesis of the DNA that will be continuous, leading, and there is formation of leading strand. So in that case, in this case, there is the one will be a leading strand, another one will be called as lagging strand. See, in that leading lagging strand, this continuous synthesis, there is lagging strands. In this lagging strands, there is formation of the Okazaki fragment. See, this fragment will be called as Okazaki fragments. See, then there is formation of the new strands that will be complementary to the parental DNA by the DNA polymerase and organization into the chromatin structures. See, this is your DNA replication. In short, see, what is the features of this DNA replication? DNA replication begins with the unwinding of the two anti-parallel complementary strands, and this complementary strands will be formed into the new strands. See, in that case, there is the code that will be found. This code will be in the form of ATCG. These are the nitrogen bases. This nitrogen bases is the one type of the information. So this information will be called as genetic information. That genetic information that will be transferred from one generation to the another generation means the hereditary characters that will be depends upon on this. See, this one will be called as, this is the DNA, and when this one will be unwinds, there is formation of this that will be called as 4. This is called as the 4, where one strand that will be continuously formed. Like this one will be called as leading strand, and the one there is formation of the patches. Means there is formation of the new strands in the form of the pieces, and this one will be called as the lagging strands. See, okay, thank you.